Welcome. Today we are going to talk about the development of embryological development of third week. In previous two videos, we have seen till the process of implantation has happened. Now in third week, first of all, the process which is going to start is gastrulation. Now what is happening? The bilaminar embryonic disc will now convert into the three primary germ layers like ectoderm, mesoderm and endoderm, which will be the derivatives of epiblast only. Gastrulation begins with the process of formation of primitive strate at the caudal end of embryo, which is now called as gastrula, and defines the morphogenesis into sides and ends and surfaces. So, if we take out that bilaminar embryonic disc previously, which was formed by the two layers epiblast and hypoblast, there you can see we can decide the cranial end and caudal end. This whole thing is epiblast. Everything is happening in epiblast only. So, first of all, there will be a formation of primitive streak. Primitive streak is a uh, plane of medium which is formed by the proliferations of median migration of epiblast length by addition of cells from caudal to cranial end. Means it moves towards the cranial end. The upper end becomes swollen, which is called as primitive node, and there is a small pit inside that node that is called a primitive node. So opposite to that side, the cranial end side will form the procaudal plate, which will be towards the future mouth. And on the lower side, it is the cloacal plate, which will be the future anus. So what do primary germ layers become? So ectoderm normally becomes nervous system, skin, epidermis, teeth, extra. Mesoderm becomes muscles, bones, cartilage, blood cells, vessels, skin, dermis, kidney, ureters, and genital system extra. And endoderm becomes GI tract, liver, pancreas, except islets, respiratory tract, bladder, likewise. These are just an example. There are many more, and some organs are having origin from more than one germ layers also, which we will talk later on. So first of all, we should remember this particular diagram, which is taken in the from the bilaminar disc from the epiblast side with the formation of primitive streak, the gastrulation starts. Next, we can see a small animation, how the things are going to happen or how the cells are going to move. So you can recall this is bilaminar disc where the cavity has been increased. And now you see the video. Upper one epiblast, lower side it is hypoblast. Then first of all, a small groove type of thing will form and the cells will invaginate the gap or replace the cells which are present on the hypoblastic side, and it will be called as endoderm. Then it will filled by the cells which will be called as mesoderm. The process is very lengthy. It is just an animation for making you understand how the things are going to happen. So the first layer to be formed will be the endoderm. You should remember. Then there is a formation of notochord inside the mesoderm only, which will be future part of the nervous system we are going to develop from this particular part and vertebral column. All these things will be forming from this particular notochord. The process will be known as notochord formation. Then it will be neurulation. We'll talk about them later. Next, we can talk about the stages of gastrulation. So on day 14th or 15th, you can see epiblast delaminate or migrates and displaces the cell in the hypoblast layer. You can see these cells are displacing the cell in the hypoblast cell. And the new cell, which is layer which is going to be formed, is called as endoderm. You can see upper side, there is a primitive stick, epiblast, hypoblast, and this is newly formed endoderm. This is umbilical vesicle, it is yolk sac. Next, what will happen? On 16th to 17th day, there will be groove-like formation. Then more and more cell will deviate downwards, which are red in color will be mesoderm now. Yellow color will be the endoderm. Remember, all these germ layers are developing from epiblast. Hypoblast will not take part. Remember this again and again. The epiblast then delaminate and proliferate to form a middle layer called as mesoderm. Epiblast remaining on the top of the things becomes ectoderm. And the bilaminar disc, which was previously we have seen, now becomes trilaminar. That is, three germs layer are going to be formed. Then further, what is going to happen? The mesoderm continues to proliferate and fills the middle cavity. This thing, you can see red in color, they become more and more size and completely fill the cavity. And lastly, what is going to happen? There is a development. Mesoderm that passes through the primitive node grows cranially and becomes notochord. In the first diagram we have seen, there was a primitive node which was developing on the head of the primitive streak. At that particular point, there will be some specific growth. In the center, you can see here, that will be called as notochord. The notochord is the key for organizing center for neurulation and vital CNS development. So this is how the next process has also been initiated. Now we can see notochord formation animation. You recall the things? Epiblast, hypoblast, then here there is an opening. You can see from where the things are going to come. So this is ectoderm, endoderm. This was the mesoderm. Now this is umbilical vesicle. This is primitive bit from which the proliferation will occur. First of all, there will be formation of notochord canal. Then it will be converted into the notochord proper. Again, you can see this ectoderm, endoderm. Identify the mesoderm, primitive pit or peak. Then from that part, the specific proliferation will be done called as notochord canal. Now we can see the different stages also how the things are going to be formed in the neurulation. The neurulation is the process of formation of neural tube, which is future CNS. Concurrently, major changes occur in mesoderm, forming somites and coelom. So ectoderm will go for the neurulation, forming the central nervous system. Mesoderm, there will be two things. One will be somite formation, then coelom formation. Somite formation will give rise to the axial skeleton and associated muscles and skin, dermis, everything. Coelom will form the body cavities like pericardial cavity, pleural cavity, and peritoneal cavity. At this stage is very important because the neural tube is going to be formed, which will convert it into the future CNS. So at this particular time, the rapid cell process pregnant women are recommended to take folic acid supplement to support DNA replication and decrease the chances of neural tube defects. If the neural tube is not completely closed, then there are some anomalies which can happen like cranial defects. It is encephaly, and caudal defects. It will be spina bifida. So this is the importance of the neurulation. Now we'll see the neurulation stages. So you first see the diagram, that is ectoderm, endoderm. Then now mesoderm is divided into three parts, which is just near to the central. It will be paraxial, then intermediate, and then the last will be lateral mesoderm. And from these three different structures or body are going to be developed. Then you can see a neural plate development here on the upper side, and this is notochord. As previously we have seen, the notochord induces the overlying ectoderm to become thick and form neural plate. Meanwhile, the mesoderm proliferates to form a thick longitudinal column of paraxial mesoderm, continuous with intermediate mesoderm, and thinning into the lateral mesoderm. Means the mesoderm is subdivided into three parts. Next, what will happen? Now the neural plate invaginates in the midline, creating a neural groove. Groove means a linear depression. Small cracks form in the lateral mesoderm called as coelomic spaces. You can see these are coelomic spaces are developing. This is again paraxial, intermediate, and lateral. This is notochord. Now neural groove, you can see a small linear depression is developing, and these were the neural folds. Next, the neural folds which were developed, they will become more deeper and deeper, forming more deeper neural groove. Rest is same like, but the names will be changed. Like the paraxial will now give rise to the somites. The coelomic spaces coalesce means combined together to form a horseshoe-like cavity known as coelom. There are three body cavities which are going to develop. The paraxial mesoderm now divides into paired blocks called as somites, expanding in the both cranial and caudal direction. They will give some specific development in the head and neck region specifically. Then there is lateral mesoderm, intermediate mesoderm, neural groove. All things are present likewise. So as a last stage of neurulation, somite and coelom formation on the 19th and 21st day, the neural folds fuse to form a neural tube below the surface of ectoderm, while a special group of cells migrate from the neural crest tube. You can see some neural crest tube. There are some cells which are separating from it. These are called neural crest cells, and these are some cells which are important, have important role in certain parts of our body. which are actually mesodermal or endodermal in origin, but has some neurogenic effects also, like adrenal glands or adrenal medulla, especially where chromaffin cells are present from the neural crest, and they act like a adrenaline and non-adrenaline, that is sympathetic and parasympathetic hormones. The coelom separates the lateral mesoderm into the somatic mesoderm, body cavity, and especially mesoderm, that is gut. So this is all happens in this particular week, that is from late week 3 to early week 4. The formation of neurulation is going to be completed. Neurulation simply means formation of neural tube, and this diagram is very necessary. You can recall from here, there is coelom are going to form, notochord, ever to notochord, neural tube, neural flesh are present, then surface ectoderm is present, mesoderm intermediate and part is present, somatic mesoderm is present, splanchnic mesoderm is present, and it is separated by a coelom. So next we are going to learn about the week 4 in the next lecture.